Okay, so now we have an RNA molecule that is very similar to our DNA molecule. Not exact, but very similar. So using the sequence of nucleotides in the DNA molecule, this RNA molecule was built by this RNA polymerase. And it's not exact, it's not quite as stable as a DNA molecule. Two nucleic acid DNA nucleic acids bonding together. It's much less stable, so that it comes off the DNA molecule, and the DNA can zip back up again on this single-stranded DNA that has this same sequence of nucleotides can now go outside of the nucleus with slightly different nucleotides with this uracil now in place of a thymine. Okay, so before it can actually go outside of the nucleus, right, we need it, introns need to be removed. Let me get my pen. From our RNA molecule. So these introns are now cut out, and exons are now spliced together. And what helps me to remember exon is that the exons can now exit the nucleus. And before it can exit, a cap is put on one end, and a poly A tail is put on to another end. And this helps in the process of, let me go back, the process of translation. Okay, we'll see that in a minute. Let's take a look at this picture. It's pretty cool. So this is an electron micrograph of a DNA molecule right here, and coming off of it there are these strands right here. So this this is the DNA molecule, and along the DNA molecule there's RNA polymerase, and it's transcribing the DNA into RNA. And that's what these strands are right here coming off. Those are the RNA molecules. So what do you think? Looking at this molecule, which direction are the RNA polymerases, because there's many of them, transcribing DNA into RNA? So there's an RNA polymerase here, RNA polymerase here, and here, and here, and here, and here. Yeah, it's going in that, wait a minute, make sure I've got it right, and going in that direction. Yes, because it's going, as it's going along, our RNA chain is getting longer. And so these are much longer, these chains right here. So it started back here, probably the promoter was back here, and going along, going along, going along, and this one kept going, and when this one was going, another RNA polymerase hooked on and started transcribing, and then another, and then another. So these RNA polymerases out here have been going the longest and have the longest chains. Here's another picture, and so we can see transcription started back here, and the longest chains are this direction on our DNA strand. These right here are all ribosomes. So what do you think? Is this a prokaryote or a eukaryote? It's a prokaryote because the ribosomes and the DNA are in the same place. In eukaryotes, the DNA always stays in the nucleus, and the ribosomes are made in the nucleolus, but then go straight out into the cytoplasm through the pores, where translation happens outside in the cytoplasm. So what's interesting is here transcription and translation are both happening at the same time. So these are all ribosomes that have hooked on to our RNA molecules and making proteins right away. So you can see, I mean, even as our RNA polymerase is going along transcribing, the ribosomes start right away making the proteins. So bacteria can make proteins extremely fast because all this can happen at the same time in our nucleoid region of the bacteria. Okay, so let's go back to our eukaryotes. So what's happening in our eukaryotes? Okay, so the RNA molecule that was made through transcription can now go outside of the nucleus in our eukaryote through the pores. And the pores are special proteins that allow the RNA and the ribosomes that were made in the nucleolus to leave the nucleus through facilitated diffusion. So no energy is needed, but there does need to be a concentration gradient. So there's a higher concentration of RNA in here, lower concentration of RNA out here. So RNA goes out into the cytoplasm where translation happens. Okay, so let's get a closer look at what's happening here with our ribosome. And these molecules right here are tRNA molecules. Okay, so this big molecule right here, actually there's two subunits. There's this big part right here, and then there's this littler part right here. These are subunits of our ribosome. So this is our ribosome. And remember, ribosomes can either be floating around in the cytoplasm or they're attached to our endoplasmic reticulum. Okay, so our mRNA molecule mRNA. So M stands for messenger RNA. In transcription, there are many different types of RNA molecules that are made. We're going to look at 3 of them. One is this one, the messenger RNA. Messenger RNA. The other one is this one right here, which is our tRNA molecule, which stands for transfer. And then there's also another called an rRNA molecule, and that's the ribosome RNA. So this molecule right here, our ribosome, is made up of our RNA and proteins. Okay, so we've got all of our RNA molecules. Here's our ribosome. Our messenger RNA has just been transcribed in the nucleus, has come out, hooked onto a ribosome. Let's see. I kind of think of it like a docking station, that it docks right here. It's called a binding site. So there's a site right here that the messenger RNA binds with or docks with. Okay. So this molecule right here, our tRNA molecule, and this is an RNA. It's a single-stranded RNA, so it has our backbone of sugars and phosphates, and it also has our nucleotides in here as well. And it's folded up into a transfer molecule. And what it's doing is it's transferring these right here. These are the amino acids. And it's transferring the amino acid to the ribosome so that the ribosome can then covalently bond each of the amino acids together. Okay, so how does it know which amino acid should go where? So this is where we finally get to the point where we can figure out how these complementary base pairs hold the code for these proteins. So these three nucleic acids, three nucleotides are called, together they're called a codon. And one codon codes for a specific amino acid. So we have, let's take a look at, let's take a look at these three right here. So guanine, uracil, and cytosine. In our mRNA molecule, this codes for an amino acid called valine. So on our tRNA molecule, there are three nucleotides that are sticking out at the end of this molecule, and they are complementary to the nucleotides that are on our mRNA molecule. So we know that cytosine bonds with guanine, we know that adenine bonds with uracil, and we know that guanine binds with cytosine. So this tRNA molecule is just floating around in the cytoplasm, and it comes in, and because they're attracted, it bonds in and sticks right there, and then moves down, and this amino acid is going to be bonded to that amino acid. Okay, so In the next video, we're going to talk more about how this happens.